very well-known biomolecule, the novel coronavirus spike protein. Infamous, really. So just take a minute to see if you can make any sense of what's going on with this depiction. I know it always looks like a jumbled mess at first, but what can we at least observe? By the end of this lecture, we hope to make even more sense out of it. But so you might notice, okay, I see a lot of these sort of fettuccine noodle looking, these flat noodles. And then I also see these spirally looking depictions. So these are all really cartoon sort of ish depictions because we don't see the individual atoms. There's just too many. It makes it kind of busies it up. So we're looking at the folding, right, of the overall structures, not just the primary attachments. We'll also be able to interpret another, at least to a greater extent, figure from a paper that first discovered it. So this is more detail about the structure. You might kind of have some intuition you didn't realize you had about what this all means. So this is actually the amino acids as they're connected, but they're letters. Have you seen that before? And there's alphas, betas. What does that all mean? And then there's just a component of the structure, and they're indicating they did a 180-degree turn, so we can look at it more closely. So we'll get right back to this by the end and look at it more closely, hopefully with a little bit more knowledge under our belts about that. We're going to start off from the beginning, the very basics. We will accelerate quickly to that figure. So going back to basics, early high school biology probably, you've probably memorized a definition. Proteins are made of amino acids, right? So here's a polypeptide. I've heard the word polypeptide. What does that mean? Poly, many, peptides. Many amino acids linked by peptide linkage. So that's the linkage that is made to connect them, the amino acids. So let's find the individual component amino acids. If they're made of amino acids, that means it's a repeat unit, it's a building block. And we've seen cartoons where they're little blocks stuck together. Now we're looking at what those blocks really are. So let's see if we can identify these blocks and redraw them. So I'm going to circle all the unique things, just like I would in any polymer, until I get to a repeat. So notice I have nitrogen, carbon, carbonyl. Nitrogen, carbon with an R group, carbonyl. Nitrogen, carbon with an R group, carbonyl. So we see a pattern there, right? So I return to nitrogen, and that's when my unit is over. So let's connect these. So nitrogen, carbon with an R group, carbonyl. Nitrogen, carbon with an R group, carbonyl. Nitrogen, carbon with a carbonyl group, carbon with a R group, carbonyl. It's getting to be a tongue twister. Nitrogen, carbon with an R group, carbonyl. So now it looks a little bit more like those, pictures in the books where they have the amino acids are these building blocks. That's what's inside those boxes. So that is a polymer. Let's draw the repeat unit, aka, instead of drawing 4 units, depict the polymer with only 1 inside the brackets. So we're going to draw brackets, and we'll say, okay, that bond to the squiggly line is where it starts repeating. Nitrogen, carbon with an R group. Notice that's a stereocenter. Carbonyl. Then I repeat, so my bond exits the brackets, and I have N for however long my polypeptide happens to be. Let's draw the monomer by itself before it made those connections. So the monomer is a primary amine most of the time, except for one of the amino acids, and it has a carboxylic acid as well, so instead of that carbonyl. So I'm going to change that nitrogen to a primary amine, and then I'll have my carbon with an R group. Notice that stereocenter once again, and instead of just a carbonyl that makes an amide eventually, I'm going to have a carboxylic acid. Okay. This is called the primary amino acid, carboxylic acid. Now this side chain is on what we're calling the alpha carbon again. It's alpha to the carbonyl. On the alpha carbon can vary. So we can have R will even equal H in one instance, and then it will equal anything methyl, phenyl, or benzyl group actually, indole. So lots of different groups, R groups could be put on that template, but we're going to max out at exploring the 20 standard amino acids. When you look at these 20 standard amino acids, you will classify them. There's a lot of ways to describe them. Basically, looking at their structure and deciding, is it hydrophobic or hydrophilic? That often has to, in some books would phrase it as polar versus nonpolar, right? So hydrophobic or hydrophilic, aka polar versus nonpolar. You might also classify it as acidic, basic, or neutral. In other words, neither. Non-acidic. It's not, because neutral just means it's not charged. So non-acidic is a better term for that, or non-basic, because it's not reacting as an acid or base. The amino acid part of it itself, amines, we know act as nucleophiles and bases, and the acid can donate the proton. So other than the peptide linkage forming, which is when the amine acts as a nucleophile, we can have an acid-base reaction happening. So I'm going to draw that product. So what I'm going to do, in other words, is take that structure right here and draw what happens when the amine acts as a base, picks up an H. Who does it take the H from? From the acid. This loses the H. So I'm going to draw this with the extra H and this with the missing H. So NH3, ammonium, still connected to the R group on that carbon. Then we have our carboxylate. So this is called the Zwitter ionic form. Which is preferred at physiological pH. So dominant physiological pH. So that's equivalent to writing it out the way we did before or here. So it just depends on what you're looking for, whether you want to draw charged or neutral. I'm going to show on the following chart, they're all going to be neutral, but you could have just as easily written them out as the Zwitter ion. So here are the 20 standard amino acids. I'm going to give you a chance to digest that. We're going to try to get comfortable with them. I think memorizing them is probably in your future for most science majors. It's health aligned anyways. That happens. And it doesn't hurt to be able to do the problems more quickly because you're really familiar with them. So I'd like to kind of think of them as more of a story of 20 friends and relatives. They are related in some ways. And so there are countless mnemonics out there because memorizing is a very common thing in some of the later biochem courses. But for me, the problem is remembering the mnemonic. Was it like my shoe fell down? And what did that mean? So what I'm going to try to do today is share with you connections related to the organic structure that can help you remember their sort of familial relationship. Whether you count that as a mnemonic or not is fine. But some things to note, because we're going to also categorize them. So they could be arranged in lots of different ways, whether they're polar or non-polar, acidic, not acidic, non-acidic. Otherwise they don't have this acid-base property. And so books might have charts with them crossing different boundaries. especially the borderline ones. I took this one from Garrett and Gerritsen, but it's not, it might have a few out of place with another book. So wherever you go in the future, if you use this page as reference, that's my disclaimer, be ready to adjust it according to the preference of that author. That's just for these categories. So in other words, if we categorize it by its properties, not acidic and hydrophobic, we could see why. So notice, here's our amino, here's our acid. I'm going to circle. Now there's no R group drawn in, it's specified. That's what dictates what it is. They all have this in common, amino acid, amino acid, amino acid, amino acid, but the middle carbon has a different R group. So that side chain tells us what amino acid it is. So I'm going to go around and circle them. Glycine, R equals H. Hence it's not a stereocenter, achiral. But then alanine, R equals methyl. Valine, R equals isopropyl. Okay, so we'll be looking closely at all of these. And we go on. Leucine has an extra carbon than valine. So it has a CH2 than the valine. And valine you kind of remember easily because that isopropyl is kind of a V shape. The leucine is, I've always used to think of it as kind of like a older sibling to valine because it's longer. It's got one more carbon. Otherwise, it's a similar structure. So, you might think of valine as like the little sister Valerie or Val, which is a common name for shorthand for that name. And this could be Lucy, the older sister. And then isoleucine is an isomer of Lucy or leucine. So notice that the CH2 has the same number of carbons, but the CH2 is taken out. And so it's connected straight back to valine's shape and then added over here. So it's kind of like the fraternal twin that has a one leg longer than the other, if you will. Anyway, so those are all related by that relationship. And they're also all nonpolar or hydrophobic, right? More hydrophobic ones are proline. Notice proline has a very unusual R group. It's a ring. So the R group is also attached back to the amino part. It's the only one that does that. There are other ring side chains, but this is the only one that integrates the amino part into the ring. And so like I said, other rings include tryptophan, which has a CH2, but then it has this big fused ring system. You might not remember this. The name of this is called an indole. When you have a benzene ring fused to a pyrrole. So in a pyrrole is similar to what we see in proline. That's kind of where it gets that name. Phenylalanine. An organic chemist would simply call this benzyl, right? A benzyl group, a CH2 on a benzene ring. But we could call it phenyl. So if you cut it off there, there's the phenyl. And then the alanine part. Where do we get the alanine part? So alanine, I'm going to circle in yellow so we don't get too much busyness going on here. Well, alanine is this guy. So those are related. It's named after alanine because it's one carbon to start the side chain and then a phenyl attached. So phenylalanine. So by that token, I would have really liked it if instead of tryptophan, they called it indolealanine because that would follow that same logic, but you can't have it go that way all the time. And so the R group is here. Yeah, but the alanine that I circled in yellow repeats, hence it's in the name again. And methionine, also nonpolar. Here's our side chain here. CH2, sulfur, thio, that's the thio part. Meth comes from the methylated sulfur as opposed to SH. So it's methyl sulfur, methionine. Thio is that old sort of root word for sulfur. I'm actually going to highlight these just so we don't get too bogged down by, so let's make them, let's take a little coloring break. Might make this a little fatter to go faster. So the indole with a CH2, methyl, valine, leucine, isoleucine, phenylalanine, Methionine. Okay, those are our hydrophobic side chains. They also are not acidic. Now, if the reference book you use classifies them as acidic versus basic, the only acidic hydrophobic side chain would be the one with sulfur, although Klein doesn't classify it as terribly acidic, so it's, you know, kind of borderline. And I noticed I actually have an error that I thought I fixed, so I apologize. I have an extra carbon, so I'm going to redraw this for you here. So cysteine, which is a really well-known one, it just has a CH2, then the S directly attached. Okay, so cysteine. Let's highlight the side chain. CH2SH. Okay, so hydrophilic side chains will make up the rest of these, and then they're going to be classified as not acidic compared to the rest, more acidic or just purely basic. So notice what they have in common. The ones that are not acidic are alcohols or amides. The ones that are acidic have carboxylic acids or a phenol. The ones that are basic have an amine of some sort or heavy on the nitrogen. Okay, so let's go through those names and also highlight what's going on there. So here's, oops, I'm going to draw on our side chain. So methionine, CH2SH, is a lot like serine, which is CH2OH. And then threonine has an extra methyl. Serine, some of the history of the names is not quite so helpful with memorizing. Like serine is named, a lot of them are named from where they're first isolated or discovered. So like serine was isolated from a protein that was used to manufacture silk. So during silk production, they discovered abundant amounts of serine. So it's named after the Greek root for silk. It doesn't really help you remember the structure, but it's cool history nonetheless. Threonine is so named because it's kind of similar to the threose, which would be a sugar with 3 carbons. And why would we say that? Because it has 3 carbons as part of the coming off the alpha carbon, as well as an OH on one of them. But I mean, it's really not a sugar, it's an amino acid. Here is the side chain here. We have an amide with the CH2. And then, so that's asparagine, and asparagine comes from the fact that it's found in asparagus. Glutamine is found in gluten. Glutamine is related to glutamic acid. So glutamine is the amide. Glutamic acid is also found in gluten. Same structure, CH2CH2, carbonyl, but instead the last part changes. OH versus NH2. So amide versus, so these are related. Whoops. These are both found in gluten. Found in gluten. And knowing that they're found together is helpful because with their similar names, glutamic acid helps us think about the fact that they have this piece the same, right? And then the part that's different, so this is in the glutamic acid, the part that's actually different is here. So amic acid comes from the fact that we have a carboxylic acid. Amine comes from the fact that in the green circle we have an NH2 instead of the OH. We find a similarly striking similarity between also the structures of asparagine and aspartic acid. Aspartic also is found in asparagus. Those are both isolated first from asparagus. So I'm going to go back to highlighter mode. Look at that structure. Carboxylic acid, CH2, as opposed to CH2 amide, right? So in other words, we could do that same thing. Aspartic acid, I want to go back to pen, sorry. Ic acid indicates we have a carboxylic acid. And then the -ene ending is for the amine. The rest they have in common. So it's a CH2 with a carbonyl, a CH2 with a carbonyl. Right? So that's the aspar part that has in common. Okay, so this is found in. Doesn't mean that asparagus is the only thing that has it. It's just where it was first discovered, so it gets named after that. Okay, finally, the last acidic one has a phenolic group. So no surprise it's acidic. We know that's a pretty low pKa compared to any other oxygen alcohol group. So that's tyrosine. CH2, benzene ring, OH. So there's a lot of side chains that are aromatic or that have that ring system. I remember TA telling me once, like, think about it as a tire because it has that O, whatever it takes. So the tyrosine, because the TYR doesn't really relate as much to the particular structure. Okay. Basic ones, we could see why they're basic. They're heavy on the nitrogen. Let's highlight in blue just the side chains. Highlight. CH2, CH2, CH2. There's 3 CH2s and an N. Lysine. Notice that we do that again. CH2, CH2, 3 CH2s and N. And we replace one of the H's with that bigger group and we get arginine, right? So lysine and arginine are similar in that way. Let's see, the history I have noted for arginine is also not as helpful with the name because arg is kind of comes from that root for silver, and I guess the crystals were kind of silvery, like Ag on the periodic table, hence the strange symbol for silver. Histidine also comes from the Greek root for tissues because it was found in tissues, so it's not very helpful with the structure to help you remember what's going on, why we have this heterocycle. So some of these you will just have to remember, but it helps to remember them. They're basic, so the nitrogen heavy ones, if you know which ones are basic, then you can kind of help flesh that out. Now, a couple things we want to also, we're going to spend quite a bit of time here. A couple things we also want to be comfortable with. The stereochemistry. What did you notice about the R group that's on the carbon? So I'm actually going to write a little note up here. So there's 20 standard amino acids. Of the 20, 19 are chiral. So that means one is not. Which one is that? That's glycine. So glycine's not chiral. That's because R equals H, and there's already an H on that carbon. Therefore, no stereocenter. So all the rest are chiral. They are all described like sugars. There's a system for naming them. They're stereoisomer. They're all described as L. But we could also assign R and S designation to them. So 19 of them are L, but only 18 are, so most of them are S. So 20 chiral. Glycine can't be L or R or S because it's not chiral. But of the 19, all are L, but only one is not S, right? So all the rest are S. How could something be L but not be L and S and something else be L and R? So we have to look at that convention for how they describe this stereochemistry. So the L and S, the L and D and L configuration is very similar to what you would do with to analyze a sugar. So what we have to do, I'm going to start off with a simple one, and I built a model. I encourage you to. So I built alanine, the first one that we encountered that has a stereocenter. So right below the space, I'm going to draw alanine again. So Alanine has the NH2, carboxylic acid, carbon with the R group coming out at you. So I'm going to draw it that way so it's coming out at us. So nitrogen's usually blue, so that's, you watch the blue one. So here's alanine. We have a wedged methyl. I'm going to write methyl here, and H going back, and then I'm going to write CO2H just to kind of make that a little easier and condensed. Okay. The convention for writing this in a Fisher projection, yes, it goes into a Fisher projection, is that the CO2H is always the head. The tail is the side chain. And then you determine where the H and the NH2 goes. That equals H or NH2. If the NH2 is on the left, when you put it in this orientation, we have an L sugar. I mean, L sugar mode, L amino acid. Okay, so we want to convert this to a Fisher projection, but notice how this is drawn. This is just fine to have this as the head, but my tail is not allowed to be the nitrogen. It has to be the side chain. So I need to rotate this so that the side chain is in the plane where my feet would be, and my nitrogen is coming out of the plane, right? So here, I'm going to let this methyl come out towards you. Here's the nitrogen. The nitrogen's not allowed to be in the plane. It has to come out of the plane. This is supposed to be in the plane. So that's good. That's where the head's going to go. So this is where I would look down and put my head there, and my feet would go there like this. So this is good. We have to leave it the same. This is the part we need to rotate. Okay, so nitrogen can't be in the plane, has to come out. So what do I do to get it out? I come forward toward you, right? R group is still not in the plane. Needs to be at my feet. Got to go again. I could have so now we're good. I could have conversely, let's go back. Could have conversely saved myself a little rotation if I just went away from you this way, right? So my R group, so I like to think of the frame of reference of the R group. Where's the R group need to be? It needs to be in the plane, not out of the plane. It needs to be a line, not a dash or wedge. Rotate it down so that it's a line. Here's my planar framework. These are out of the plane. Okay, so now the nitrogen is on a dash for you, right? Let's do that. Okay, so I'm going to rotate my nitrogen into the plane so that the out of the plane so that the methyl is in the plane. So in other words, I need to go like a little propeller. The methyl goes down to the line, nitrogen goes down to the dash, H comes out to the wedge. So let's show this transformation, or rotation rather. It's a new conformer, CO2H. If you did it right, you have the side chain right there, methyl. And then fill in your dashes and wedges. So H came forward, NH2 went back. Now we will look at the amino acid to draw a Fisher. Where do we need to be? We need to be right here because the carbon's going up in this case. So we are going to look from below, from above, excuse me. Let me draw my eyeball. Okay, look down from there. And when you're doing that, right, get in there, get in that plane. Your head's at the car, your head's here. The methyl's down below. They need to hug you. you should see on your left is the amine. So let's fill that in. CO2H is the head, methyl is the bottom. Our one stereocenter has amine on the left, H on the right. Because amine's on the left, this is the L version of alanine. So we can quickly convert this one into a Fischer. Let's see, this one. Because we have the side chain not drawn on a dash or wedge. Why in the world would we draw it the other way then? So why would we do it this? Why is that whole list have them drawn this way when that requires me to do a rotation to get into a Fischer projection? Well, first of all the naturally occurring amino acids are L, and so that's something that would be understood no matter how you rotate it. It's useful to kind of see the consistent parts on the plane and only vary the one wedge piece. But additionally, what if we want to assign R and S? This is the more convenient conformer, right? This is the more convenient one. Why? Because it has the H in the back, the lowest priority group. So let's assign R or S to this. So 4 is the lowest priority. Now we have methyl, which is carbon. Carboxylic acid starts with a carbon. Amine starts with a nitrogen. Amine wins, number 1. Between methyl and carboxylic acid gets number 2, and the side chain gets 3. We sure look like we're going counterclockwise, all right? So this is S. So the majority of the amino acids that are chiral are actually S, because the side chains will be number 3. There's one weirdo that does not fit this mold. Okay, so we've checked off how we could see that 19 are chiral. We see that they could all be L, because if you redraw all of these in the Fischer, you're going to be able to get the amine on the left with the side chain is down at your feet. But why is there, if they're all L, why are they not all S? Only 18 are S, so 1 is R. So that'll give us our 19 total. Which one is R? Has to be one that, the only difference between all of them is the side chain. So it has to be one that changed the numbering priority on the side chain, right? So they all have, in other words, every single amino acid has this in common. Every single amino acid has this in common. Every single amino acid has that in common. None of them have that in common. They all differ. So the majority of them start with a carbon bound to something else that's low enough priority and so to decide, remember what we had to do to decide who got the higher priority, the 2 and 3? We had to decide who was a higher priority, carbon with H's and another C, or carbon with 3 bonds, or 2 bonds to O. Actually 3 bonds to O because there's a double bond. So we're always picking CO, but there's one other side chain that has a bond to something with higher priority, and that messes up our assignment, our neat little wrapping up of calling them all S or R. It's cysteine. Let's assign the stereocenter in cysteine. 4, that's the easy one. 1, so because we're going nitrogen versus hydrogen versus carbon. So 1 and 4 are clearly the same as they were last time, because nitrogen has a higher priority than any carbons. Now these are 2 carbons. Let's move out. Carbons attached to oxygens or carbons attached to sulfur. Think about the Conning-Gold-Prelog system of establishing priority. Here's my periodic table. I have a puzzle periodic table. My kids just put this together. So it actually snaps together. So I was very excited by this. Here's oxygen. Right there, and below it is sulfur. I'm afraid if I take this cardboard away, it'll all break. It might stay. That's kind of cool. But yeah, below it is sulfur. So higher atomic number wins. Doesn't matter that carbon was bound to oxygen 3 times. It was bound to sulfur once. Sulfur wins. So cysteine is the weirdo. Cysteine, that sulfur messes it all up. So it's actually 2 instead of 3. So S, priority of S is higher than O. So when we connect 1, 2, 3, we're going clockwise. Therefore, we have an R stereocenter. So this is still L-cysteine, but you could also say it's RL-cysteine. So this chart has only the L amino acids drawn the way they're found naturally occurring. So our body is once again showing its extreme stereoselectivity and antioselectivity even. And so not that it's not possible to synthesize artificially the other enantiomer, that's just not what's found naturally. Now notice the little asterisk here. The stars mean essential amino acids. That means they're just not naturally made. They have to be obtained through diet. So like lysine, arginine, histidine, for example, those are through diet only. I want to draw your attention also to, eventually when you do enough biochem, you'll accidentally memorize some of these other abbreviations. So lysine could be abbreviated LYS, that's pretty logical, but then K. And why is that? It's because there's other L. Is there another L? Yeah, leucine gets the L, stole the L. So there's lots of A's also. Aspartic acid can't have the A because who had the A? Alanine has the A. I kind of remember that one because it's the first one, the first one on the list. It's like the next most simple after glycine. Cysteines is pretty obvious, a C, he's one letter. Asparagine has an N, and that's because and then aspartic acid has a D. So why does it get a D? There's some logic to it, like you think about aspartic instead of tic, aspartic acid, you can kind of remember that, right? So glutamic acid, notice, is just like aspartic acid. It goes D then E. And I relate those because they're the next in the alphabet. So in other words, D then E. Notice that aspartic acid has 1 less carbon than glutamic acid. Otherwise, they're the same. So that helps. For glutamine, I remember the days I thought of that as glutamine with a Q, just in case you have to know those 1-letter abbreviations. Arginine's the pirate, arginine. So that kind of helps as well. So those 1-letter ones are really helpful when you have a really long peptide, polypeptide that you're trying to show the amino acid sequence without having, tripling its length by having the 3-letter abbreviations, or even worse, the whole structure, right? So that could be a little cumbersome.
S? There's no S. Carol. 900.
I want you to say hello to an old friend. A reaction in which carboxylic acid is treated with an amine. And let's put a catalyst to make that a more favorable reaction, because we remember that they're not very easily prone to nucleophilic attack. So what do we get? Attack, open, close, kick. We get DNHR prime. We get an amide. So here's our carboxylic acid. Does this look familiar? The carboxylic acid reacts with a primary or secondary amine in the presence of a catalyst. The product is an amide. We did this in the carbonyl section. We used acid is a catalyst. DCC is another really common one we see thrown out there. But our body doesn't need that. It uses the most reliable catalyst. What's a catalyst in your body? An enzyme. So it makes these amides between amino acid amines and amino acid carboxylic acids. So they link them together. When you make that amide from the amino acid amine and the amino acid carboxylic acid, that is called a peptide bond. When you have a whole bunch of those together, then we call that thing a peptide. This peptide bond is sometimes synonymous with the name amide linkage for good reason, because you made an amide. Okay. Let's stitch together 3 amino acids to make a tripeptide. We have to remember what happens, and we're not going to draw the whole mechanism because we've got a lot to do, and we've covered that already. So what we're going to do instead is acknowledge what happens to make the amide from an acid and an amine. It means without OH is gone. It means one of the H's is gone. In other words, water comes off. Let's do that again. Okay, so if we link them in that sequence, let's draw the resulting amides, peptide bonds, in other words, amide linkages. And we're going to leave the rest the same. H2N. Carbon up means R groups on a wedge. Carbonyl, connect to, no longer OH, it's now an N because we made an amide. Carbon is down now instead of up. Uh-oh. Our side chain, is it on a dash or wedge for serine? You better put that on a dash because it's not up anymore. Check your stereo center and make sure they're the same to confirm that. Carbonyl connects to N to make an amide. So there's our amide. N kept 1H. Now it has a carbon up. Phew, carbon up was already up in the valine. I get to leave it the same. Wedged, isopropyl, side chain. End on the acid. Started with an amine, ended on an acid still. Okay, so we would say this is a tripeptide that has 3 amino acid residues. Okay, so how would we name that? First of all, we used alanine, serine, and valine. So do I call it aliserval? Or do I call it valser-ala? What if I flipped it around? Would I then call it valser-ala? No, because flipping something around doesn't give it a new name. Just like if you turn around, you don't get a new name, right? So flipping that around, it should have the same name. So we need a consistent way to name it. So how does that happen? You start naming at the N terminus, as you probably could guess. That means where the amine is. We end naming at the C terminus. As you might be able to guess, that's where the carboxylic acid is. So it's already set up in our left to right reading order, which may or may not be easier, depending on what you're used to, but we're going to start at N, and we go in order. Ala, ser, val. Ala, ser, val. Now, if you were asked to draw what this looks like at biological pH, I'm going to leave that for you to do. You know what to do. You draw that thing all over again, except as the zwitterion. So I'll make that your own activity. Draw your zwitterion. Okay, so when we get to the amino acid sequence, okay, when we are listing them in order and naming them, we have a peptide, and the peptide has, we're going down to the detail of saying it has alanine, serine, valine in that order. Then we have described the primary structure of a protein. And you might have memorized that definition without really fully realizing what that means. So the primary structure of a protein is the amino acid sequence. Well, now you know how to name it, right? We're going to, of course, branch out. What if that's primary structure? What's secondary? Let's get there. Let's look at this is on our way to secondary now. In order to understand what's going on with the secondary structure, we have to know the conformations that it can adopt. So the primary structure is in place. We know they're all connected, but then it's about what conformer is preferred. And there's a lot of ways for it to rotate. There's so many, it's hard to believe that there's a consistent pattern, but there is. And so we'll look at one reason why we can see a consistent pattern, and it goes back to stuff we actually know. So let's draw the resonance structure of an amide, and we'll see that because we can do that, it is therefore conjugated. That lone pair that was drawn in for you is there for a reason. Conjugated. Conjugated means that the p orbital here overlaps with this p orbital in order to ever make this pi bond, that it must stay planar for that to happen, to delocalize those electrons. So that helps. That's restricted rotation right there. We're going to leave that fixed. So if you're a chemist, a protein chemist or biochemist trying to decipher the secondary structure, which we're getting to, but has to do with the conformations, you would probably leave this bond alone. It does not like to rotate for this reason, because we know we don't rotate around pi bonds and it has partial pi bond character. So I'm gonna say this has restricted rotation. Okay, so that allows it to be planar because of that conjugation. But if it did rotate, what would it look like? So let's draw our loop-de-loo arrow. We would rotate around where that pi bond would be, but it takes a higher energy barrier to get that to happen. I'm just going to leave the left the same. I'll rotate the right. So the H is up here, and now the squiggly line is down there. So what does that mean? That the squiggly line is where the rest of the peptide is, the big stuff. So let's highlight it. Now looking at where the rest of the big stuff is, how would you describe the relationship of it? We're rotating around a single bond. Yes, it has partial double bond character, but we're rotating around a single bond. We're going to call that S, S cis. Those big groups are cis to each other. Whereas here we'd call this S trans, which happens to be drawn for you because it is the more stable one. Because you want those bigger groups to be trans, right? And I think we've already answered this question. It is not easy to interconvert between them because of that conjugation. Hindered rotation, we would call it. Still possible, but it definitely limits the way proteins can fold. Otherwise, they would take on just millions of different shapes. So, for example, we're going to expand our view past the peptide bond now. Look right here. So we're going to say that has hindered rotation. I'll put an X through it. It's not happening. But look at all the other ones that can rotate. This can rotate. This can rotate. That can rotate. Too many. So we're not going to count, but a lot. You can do the math, but we don't need to. But reactivity is determined by structure, right? So the biologist would say function is determined by structure, that structure-function relationship. In the chemist terms, function is the reactivity. So structure. So we know that proteins, and so here's kind of a conundrum. We know there's all these different ways it can rotate because they're all single bonds except for that one that's hindered rotation. But if it needs to have a consistent function and it's reliable, right? React in a predictable way, in other words. It needs to have a single, reliable, stable conformation. How does it accomplish this when there's all those rotations possible? We know that single bonds rotate rapidly in solution. So here comes the solution. the protein interacts between other portions of the amide backbone in order to restrict this moment. It forms hydrogen bonds. So this is called the secondary structure of the protein. When the backbone, the protein backbone, interacts with other parts of the backbone. Okay, so i.e. backbone, H bonded to backbone. It folds back on itself, right? So you have this big, long protein, but it can fold and interact with itself. That'll keep it from flip-flopping to too many different conformations and changing its structure. So when we're discussing structure in that way, we're going to talk about, I guess I would say 2 regular, but there's 3 that I'll mention the names of. So I'll call it 3 common structures. The first one, alpha helix. Alpha helix is what it sounds like, a helix. So if you look down the center of an alpha helix, so it's helical. Here we go with the helix again, like DNA is helical, right? If you look down the center of alpha helix, the spiral moves down. Let's look at that. Imagine you're going down. You're looking at, you're following the spiral. So what direction could it go? It depends on the chirality, the helical chirality. So you can actually assign it a clockwise or counterclockwise. And then the side chains are going to point out, right? So think about what this alpha helix is here. You know, if you might need to zoom in and take a good look. Which way is that one going? Helices can go clockwise or counterclockwise. What is it? Which way is that one going? Think about that. So the side chains are pointing out. Now, if I take that view, if I look down the core, and my view would suddenly look like that. So in other words, I take that helix, that side view, and I look down, this is what I would see, the figure where we have the core more visible. And these are all the side chains pointing outward. When you have that shape, you can actually see it act somewhat like a spring. And that might have to do with how it has to fit somewhere or bind to something or be a receptor. Another option for arranging them, and this depends on how the side chains are, what side chains are present, right? So we could have a beta helix as well, but alpha helix is common depending on what's going on in terms of the clockwise or counterclockwise. Are you ready for the next exciting secondary structure? Fettuccine noodles? Beta plated sheets, actually, the more technical term. So when does this happen? How do we get those? 2 or more amino acids align. They form a hydrogen bond. Okay. I would actually say within a peptide residue. So I'm going to add that here. So within a polypeptide, the amino acid residues align and form hydrogen bonds. So in other words, instead of coiling, they align in a linear fashion. This is called a beta pleated sheet. So here's what the sheet looks like right there. Pleats mean, you might have seen like pleated clothes. So you could have a crease in the pants that pleats, or dress pants or skirts and things. So that's the term pleated because carbon, reminding you that we have tetrahedral carbons at play here. So they're not perfectly flat, but they're somewhat rigid because they're holding 2 strands in place. And then it makes this high surface area looking part of the structure, hence the like flat or fettuccine noodle, right? So Zooming in, what is happening there? We see our hydrogen bond. Let me erase that. Donors and acceptors at play. So look, amide NH bonding with carbonyl oxygen. Carbonyl oxygen with amide NH, and so on. Every time you see that sees another one, they interact with each other. So those backbones are interacting. We're not paying attention to what the side chains are doing in this case. We're looking at the backbone. So that's why they're not even drawn. When the N termini, so you find the N terminus, when they're on the same side, we will call it a parallel beta plated sheet, but if otherwise, it would be anti-parallel. And this allows them to be rigid when they have these long series of intermolecular forces. So I got a problem for you. Take that example. Is that parallel or anti-parallel? Give it a try.
Yes, no, maybe. I don't know. Can you repeat the question?
What did you find? Here's the answer. Want to see how? Okay. First, remember what we have to do? The criteria is the N termini. Are they on the same side or not? So find the top chain, look for the N terminus. All right, well, how do I do that? Okay, so this is a carbonyl. I'm going to stop when I get to a carbonyl, because that's where the C terminus is, or an N. Stop at the first atom that gives me a carbonyl or an N. So there's a carbonyl. This must be the C terminus. I better have an N at the end of this. Otherwise, I'd be wrong, right? Okay, I have the side chain I'm not worried about. I'm looking at this. There's my N terminus. Okay, let's likewise find it on the next one. First thing I circle is the N. So that's the N terminus. And I end on a carbonyl, C terminus. Don't worry about the side chain. Okay. Notice that they are not going the same direction. So it's anti-parallel. Easy enough, right? Sometimes you see that indicated with directionality, an arrow going one way or the other. The third way, so we have alpha helices, we have beta pleated sheets, then we have everything else. That's just called random coil because hydrogen bonds form in a different way, various regions. And so it's not exactly random, it's just harder to describe because there's not a nicely packaged coil handed to you to look at. So that's the secondary structure. Let's look at tertiary. So tertiary structure is moving beyond that. So moving beyond the folding. It determines how the protein responds in its environment. So if it's aqueous versus fat, polar versus nonpolar, depending on what side chains it has, it will do different things. Polar side chains are quite happy in aqueous environment, but nonpolar ones not so happy. So it will fold accordingly to adjust for that environment. Just like if you went outside on a hot day, you would probably spread out to kind of not get sweaty and air out, right? But if it was really cold and windy or rainy, you would huddle up. It doesn't mean your primary structure is altered. It's just how you're folding up, right? So it's the idea of it's folding in a response to the actual environment, not in response to its own inherent structure. Okay, so in other words, hydrophilic environment, water, blood, because blood's aqueous. Your hydrophilic chains are quite happy to be immersed in that environment, so they point out to interact with it. While your hydrophobic side chains, not so much, they're going to point in. You take that same protein, put it in a hydrophobic environment, like an organic solvent, but in the body we do like lipids, right? So something nonpolar, so fats, cell membranes, etc. You're going to have the opposite. It's going to be inverted. So your hydrophilic side chains point inward, while the hydrophobic side chains will point outward. Okay, so thinking about the environment, you want to think about how that would arrange itself. That's your tertiary structure. Another important side chain interaction is the formation of disulfide bridges, which you've probably heard about frequently. And it's actually a redox reaction. Do you see how removing H's gives us a disulfide bridge, and adding the H's back gives us the thiols? What particular amino acid residue is responsible for this? It's always the same, cysteine. Okay, let's move on to the last one, quaternary structure. I really like quaternary structures. I know it sounds weird, but you get into symmetry elements with that, which is something I've always been a fan of. So we'll get to that in a minute. So it's any aspect of the structure that joins the subunits. So you have your piece. It's like a monomer, if you will. And then they associate or aggregate in such a way that you have these pieces repeating. Now, the subunits could be lots of things. So that joins two or more. The subunits can be polypeptides. They could be coenzymes. They could be carbohydrates as well involved. So you can have lots of stuff, all these biomolecules wrapped up into a repeating subunit. So let's take a look at what happens when we zoom out and look at the final quaternary structure. Here's some examples. So let's see, my typing I think got off a little bit here. So I'm gonna fix this myoglobin and lactate. Yeah, that's good. Okay, so we have myoglobin, retinol binding protein, and lactate dehydrogenase. Look at these big things. What can we see? So notice that we have protein that we're looking at, the whole piece here. What do you notice about, so we have these connections that are not defined, but we do have these coils. So these are the alpha helices. So we can spot the alpha helix here. All right. The next one I have for you to see the beta pleated sheet. And notice the arrow? It's to show you whether it's parallel or not. So sometimes they're clear, sometimes they're not, but so these are beta pleated sheets represented in 3D. Now let's look at one with alpha helices and beta pleated sheets. Now you might notice hopefully a certain symmetry to this as well. So not only are there alpha helices and beta pleated sheets, notice how this is almost like it's looking in the mirror but inverted, right? And then we see a match here that's similar and a match there that's similar. So we could kind of, around this central point, so it's more of a point than a plane, we can pass through a point and find another one like it. We pass through a point and find another one like it, only it's been flipped. That's a center of inversion. So notice this one here is going the opposite way there. It's flipped over, but it's also still there. This is here. We can't ignore that symmetry, right? See this one? Go through that point and it's there. So we have an inversion center there. Told you I like these. Get excited by the symmetry element. So symmetry, inversion center, but notice we only have a one-to-one match. So for example, I only see one more of these. I don't see one over here or over here. I only see another one like it. So that means if we have one and another match for it, we have two pieces. We have a dimer. So can we circle the unit that's repeating? So let's do this. It's hard to say which one belongs where, but I'm going to try to get only one piece of each. Right? think I didn't take any of the other part. And so there's another match of the other one on the other side that's flipped over. It's like we took that and flipped it over. And that's how they associated together. So this is actually a dimer. It has two units that are the same that repeated, right? In this case, it's a homodimer because they're the same unit. But you can have a heterodimer, you have a trimer, right? So it's a homodimer. It's just flipped the way it's represented. That's quaternary structure right there for you, okay? Let's go to one even more complicated that we looked at the very beginning. I hope this has more meaning to you now. Look at this beast, the spike glycoprotein. Let's stop there. When I look down the core, I can quite easily see that there is an axis of symmetry. In other words, a rotational axis, where if I rotate it, if I could split it into thirds, right? So this green piece could match with the purple piece, which can turn and match and superimpose with the orange piece. So it's been colored by the scientists that rendered this structure to make it more simple. That color coding represents each unit. So the quaternary structure is a homotrimer, right? So a homotrimer is depicted with help of the color coding, right? So it's got a threefold axis of symmetry that we can rotate there. So rotating it around, it's really obvious at the core. So we can rotate the threefold axis of symmetry. Notice these, as I do that, I'm gonna get my laser pointer. So these guys right here, so they repeat. So I rotate this orange, they'll be on top of the green. Rotate the green, they'll be on top of the purple. The orange piece repeats. Orange would be repeated over here where the green is. And same with the purple, right? So that is the quaternary structure. Let's zoom in into a level more detail. So back to this picture. This is one of the units that we see with the spike protein, right? Just one of them. So it's a homotrimer. Here's one of the units. We see our alpha helices, we see our beta pleated sheets, right? Now, how about the rest of this figure? You might recognize some of these letters now. Here's the primary structure, the amino acid sequence, RVQPTESIVR. So if you go and look at your list, you can match those up, right? R is actually arginine, V is valine, Q for glutamine, right? P is actually proline, the one that's part of a ring, right? So, and then it tells you whether that, it also tells you the secondary structure, whether it's an alpha helix or beta pleated sheet. So pretty cool to be able to interpret that now, right? Okay, so maybe go back and draw some of these and try to look at that video of the spike protein and see if you can see how it forms a C3 axis of symmetry, spike protein, right? So go back to that video where it has the spike protein in complete formation. So this is one subunit, the trimer. We're gonna look at that, okay? And see if you could find that repeat unit, if you were able to circle those. That's it for amino acids.